Okay, so here we have a normal protein sequence and a mutant protein sequence, and we want to figure out what happened in the mRNA that was translated into these protein sequences to cause this non-functional mutant protein. So if we look at the universal coding sequence that I have here, we can see that MET codes for AUG, TRIP is coded by UGG, Phi can be UUU or UUC. This is only ACU. List can be AAA or AAG, and this one can be GG or any other letter. That can be A, C, G, or U. And in the mutant, we have the same here, AUG, UGG, and ILI can be AUU, AUC, or AUA, and his can be CAU or CAC. So from the mRNA sequences we have, we can determine that there was an A insertion right here in the mutant strain. And because we have double U's right here, we know that this has to be AUU. And because the next one starts with a C here, we know that this has to be CAC, as seen right here. And then because the next one starts with a U and the double A's here, we know that that's going to be. Right, so here I have a visual representation of what we just figured out. We have our normal protein strain and our mutant protein strain and the mRNA sequence that translates into these proteins. So we have the normal one and then we have the mutant protein strain. And what we figured out from before is that a single A base pair is inserted right here in the sequence, which causes a whole frame shift mutation because the DNA bases can only be translated in triplets. So this causes a whole frame shift. And now we have that AUU codes for ILI, CAC codes for his, and UAA is a stop codon. So these are now silenced and not expressed. So we now have the coding sequence of the mRNA that's translated into the mutant protein, and we can see how one insertion of a base pair resulted in the truncated non-functional mutant protein.